Hello students, welcome back to a new video of Sunanda's tutorial. So in this video we will discuss about GPCR. Okay, GPCR and about G proteins. What is GPCR? GPCR means G protein coupled receptors. Okay, G protein coupled receptors. These are largest family of cell surface receptor. So it is one of the most important cell surface receptors which are found on the cell surface that transmit signal to intracellular targets. So at first they receive this cell surface receptor or the G protein coupled receptors receive the signal, and then they transmit those signals to intracellular targets via intermediary action of guanine nucleotide binding proteins called G proteins. These are mainly present in all eukaryotes. GPCRs consist of single polypeptide of multi-pass nature. They cross lipid bilayer 7 times, therefore called as serpentine receptors. That means these G protein coupled receptors, they enter this cell membrane 7 times. 1, 2, 3, 4, 5, 6 and 7. So this protein have multi-pass nature. Okay, and they cross this lipid bilayer 7 times, therefore called serpentine receptors. So there are two types of G proteins, also called as GTPase switch proteins, present linked with this GPCR. One is called as trimeric G proteins, other group is called as monomeric G proteins. Okay, these proteins are turned on when bound to GTP. So there are three proteins called G alpha, okay, G beta, G gamma present with this GPCR. Okay, these proteins are turned on when they bound to GTP. When GTP bound to these proteins, they turn on, and turned off when bound to GDP, guanine diphosphate, and turned on when they are bound with guanine guanosine triphosphate. Okay, in absence of a signal, G protein is bound to GDP. That means in absence of signal, these proteins are in their turned off stage. Signal activate release of GDP. So when signal attached to this GPCR, they get activate, and the release of GDP and subsequent binding of GTP occur. Okay, so this binding of GTP and release of GDP is favored by the higher concentration of GTP. The intrinsic GTPase activity of this GTP binding protein hydrolyze the bound GTP to GDP and PI, thus converting active form back to inactive form. So sometimes the intrinsic GTPase activity, intrinsic means inside the cell, due to the GTPase activity, GTPase means which can break the GTP to form GDP and PI, inorganic phosphate. So when GTP gets fragmented into GDP and inorganic phosphate, the G proteins again become inactivated. Okay, when they bound to GTP, G proteins when bound to GTP get activated, but when they dissociate by intrinsic GTPase activity into GDP and inorganic phosphate, they get inactivated. Okay, so here it is the GPCR and it is the G proteins in inactive state. because they are bound with this GDP. But when signal molecule attach on this GPCR, this GDP is replaced with GTP, and these subunits again becomes activated, and when they get activated, this alpha subunit get detached from beta and gamma subunit. Okay. In case of stimulatory G protein, the alpha subunit activates effector, that is for example adenylate cyclase, because this G protein is involved in enzyme activation. It is called stimulatory G protein or GS, stimulatory G protein. So in case of stimulatory G protein, their alpha subunit activate effector. For example, adenylate cyclase. For example, here signal attached with this GPCR, then G protein alpha and beta gamma all get activated, then they activate another effector enzyme called as adenylate cyclase. This effector enzyme again activate secondary messenger.